All right, we left off here and we've started with this analogy of two-part molds. And now you know that the cool thing about DNA being double-stranded is that the two strands, one of them has got like ridges and grooves that are straight and narrow, and the other side has got ridges and grooves that actually fit. Now, they're not ridges and grooves. They're actually molecules that are complementary to each other, okay? But let's reiterate how important it is that we not make a mistake. Here is a protein, right? This is a protein right here. And this protein is hemoglobin. That little green thing that shows up there, that's oxygen. And when oxygen binds to the hemoglobin, hemoglobin's the protein that makes your blood red. When oxygen binds to it, then you can see that the shape of protein, that hemoglobin, it changes a little bit. That changing of shape, that's typical of a protein. Proteins have shape and they're like little machines. Now, when this hemoglobin is working properly, you get lots of oxygen going to all of your cells. In this particular sequence, it doesn't look like anything, at least it shouldn't look like anything to you. Each one of those letters, A, V, T, each one of those is one amino acid. Remember there are 20 different amino acids? Each letter is one amino acid. There are more than 100 amino acids that make up one of these subunits in hemoglobin. And if you make a mistake right there at that one, if instead of having that glutamine, you switch it to a valine, then you get sickle cell anemia, which without modern medicine is generally fatal, okay? So changing one amino acid out of 110 can be a fatal mistake. And that's why when this DNA stuff that has got all the instructions for making this protein, we can't have any mistakes in those instructions. If there's a mistake in the instructions, then the protein is wrong. And when the protein is wrong, you could die or get cancer, okay? So let's talk about how life gets it right so much. So remember nucleic acids. There are two basic nucleic acids, DNA, deoxyribonucleic acid. It is this double helix. Now, it's easy to say, oh, here's some DNA. No, not really, okay? One of your chromosomes will be millions of base pairs big. What is a base pair? This and that is one base pair. So maybe there are, 20 base pairs here. So 20 base pairs versus a million base pairs, this is a little teeny tiny little bit of DNA, right? So, but it is the way DNA looks. It looks like a winding staircase, and it is known as the double helix. Ooh, I think that's on the exam. It's known as the double helix. DNA is the stuff that sits around in your nucleus of your cells, and it is what is necessary for your cells to be able to make the proteins that allow them to be alive. Right? The other genetic material, the other nucleic acid is RNA. RNA is ribonucleic acid, and it is very important to the story of building proteins that we're about to learn, okay? Now, this is what a nucleotide looks like. It wasn't that long ago that we were talking about atoms, okay? There's an oxygen atom, there's a phosphorus atom, there's a carbon atom, two hydrogen atoms, right? So this is a small molecule, and it's known as a phosphate. This right here is a small molecule known as a sugar called deoxyribose, and this is a small molecule. This particular one is known as thymine, and thymine is an example of a nitrogenous base. Every nucleotide is made out of these three small molecules. Every nucleotide, whether it's DNA or RNA, every nucleotide is made out of a phosphate, a sugar, and a base, right? Now, this thing, the nucleotide, which is shown schematically here on the lower right, This nucleotide is going to be the monomer, and DNA is built out of nucleotides. RNA is built out of nucleotides. All right, so let's talk about it a little bit more. DNA and RNA are both nucleic acids. They are built out of nucleotides. DNA, nucleotides, made out of a phosphate, a sugar, and a base. The name of the sugar that is used in DNA nucleotides is deoxyribose. Literally, this D right here in DNA, that D stands for deoxyribose. Oh, let's see if I can write this. I really don't think I can. So DNA stands for nucleic acid. Deoxyribonucleic acid. It's a nucleic acid. That's what the N and the A stand for. And the D stands for deoxyribo. Why deoxyribo? Because this is deoxyribose. Okay? So you will not have any trouble remembering that the sugar in DNA nucleotides is deoxyribose because it's literally right there in the name. Now, the base. This is the base right here. And those bases, when it comes to DNA, there are only 4 to pick from. And those 4 are T for thymine, C for cytosine, A for adenine, and G for guanine. Those are the 4 bases. And remember, I said there was an analogy that on one side of the mold was like a square ridge, and on the other side, there was a square groove. Well, in DNA, the square ridges are the adenines and the square grooves are the thymines. The round ridges are the guanines and the round grooves are the cytosines. They match. They are complementary to each other. They are complements of each other or complementary. That's complement with an E, not with an I, not like, hey, nice DNA you got there, but complement, meaning they're pairs or they match or they're opposites or they fit together, okay? They are the complements of each other. So you will find that in any DNA molecule, any double helix that you've got, when there is a T on one strand of DNA, there's an A on the opposite side, just like square ridge, square groove, right? And wherever there's a C on one side, there's a G on the other side. So for here, the things that you need to know, make sure you know that the 3 things that are in a nucleotide, phosphate, sugar, and base, that the sugar in DNA nucleotides is deoxyribose, that DNA nucleotides come in 4 types, depending on which of the 4 nitrogenous bases they have attached to them. And those 4 bases are G, A, T, and C. The reason that the letter T here, has got, is red, is because the RNA nucleotides are the same except for the letter T, right? Remember that DNA is double-stranded and that the 2 strands are complements of each other. There's something else I was going to say about that. I'll get around to it. Okay, so DNA is built using literally millions of nucleotide molecules, but just 4 types of them. Okay, millions of nucleotides. Some of them have an adenine as their nitrogenous base. Some of them have a guanine, some of them have a thymine, and some of them have a cytosine. Okay, those are the 4 bases. Now, I'd like you to notice that the adenine and the guanine, they're big molecules, whereas the thymine and the cytosine, they're small molecules. So here are going to be the backbones of our DNA. By the way, the backbones of the DNA are phosphate, sugar, phosphate, sugar, phosphate, sugar. And hanging off the sugar is going to be a base, okay? If it's an adenine, it's going to be a big one. And the adenine, I don't know, doesn't matter. We'll make it pointy, okay? The matching to the adenine is going to be the thymine, and thymine's little, right? Then, let me just change pen colors here. Okay, if we've got a guanine over here, it's big, and the match to the guanine is the cytosine, and it's small. Now, the reason that this is interesting is that all of the nucleotide nitrogenous bases come in two forms. They come in purines, which are the big ones, and pyrimidines, which are the small ones. I always remember that because pyrimidine is a long word, but it's the smaller nucleotide base. And purine is the smaller word, but it's the bigger nucleotide base. So what's cool about that is you'll see in any given pair that there can always has to be one big one and one little one. So if you had a little cytosine there, and if as you are building the DNA, you put a little thymine next to it, they won't fit, right? They don't fill up the space. On the other hand, if you try and put a big one, and then you try and put another big one, again, it wouldn't fit. The adenine, guanine, cytosine, and thymine are as different from each other from the point of view of the machines that are building this stuff called DNA. They're as different from each other as black, white, red, yellow is to someone who's got normal color vision. You just can't mistake them for each other, right? They are so different, and they only go together in one way. Black with white, red with yellow, all the time. You always have an adenine with a thymine and a cytosine with a guanine, okay? So why? Why do we have these pairs? Well, remember that the goal of this system is to make millions of copies of this stuff called DNA without ever making a mistake in making the copies, okay? If adenine and thymine and cytosine and guanine are totally different from each other, then there's no chance that we're going to make a mistake. So let's imagine over here, I've got adenine, cytosine, guanine, thymine, okay? And so you're going to like erase this right here so that I can't see it. And then you're going to say, okay, smarty pants, try and make the left side, try and make it the way it was. Try and make a perfect copy of the left side. I would just remember the rules of complementarity. And the rules of complementarity are, if there's an adenine on the right side, there'll be a thymine here. If there's a thymine on the right side, there'll be an adenine here. Cytosine, guanine, guanine, cytosine. Okay, so that's what it's supposed to look like just by knowing the rules of complementarity. So, did I do it right? Yes. Was it hard? No. That's what's brilliant about this particular system, is when it comes time to make a copy of DNA, it is a daunting task. The enzymes that are doing this, we'll get to what enzymes are in a second. The enzymes that are doing this, they need to take millions of these adenine, thymine, cytosines, guanines, and they're paired together. They need to make a perfect copy of it. So what do? They just separate the two sides like we did with our pot and our mold. And then they will use one side like, sorry, I got an eraser here. They will use one side like this one right here, and they will use that to make a perfect copy of this. And by the way, they're going to use this side to make a perfect copy of that. Okay, we're going to pick up here at the next video.